cell membrane acts as a body to the cell. The hydrophilic heads and the hydrophobic tails allow for the cell to move around and be flexible. The cells are made up of a phospholipid bilayer that is demonstrated through the bubbles. When the straw is put through the bubble, it doesn't pop, but allows the straw to go through. A cell membrane is self-healing. This means that when things go in and out of the cell, the hydrophilic heads and hydrophobic tails don't leave a gap where it is, but they close in the area where the object was as if it was never there. We demonstrated this through the bubbles by entering a straw in and out of the surface. Once the straw left the surface of a bubble, it closed up as if the straw was never there, just like a cell membrane would do. A cell grows when you add phospholipids around the outside. These phospholipids attach to the surface and the cell expands. When you put the straw through the bubble and blow into it, the bubble grows just as a cell would. In the origin of life, when you added phospholipids to a cell, it would grow. When doing this to many cells, it can create life if you have the right chemicals. The phospholipid bilayer of a cell will divide when split by an object. In this case, the bubbles split when the straw cut through them and grow back after. This is called binary fission, which is a form of asexual reproduction for the cells. The asexual reproduction is a key factor in the origin of life. This is how cells reproduce without any help. Within a cell, there are many compartments, such as the mitochondria, that help organize the cell. We show the compartmentalization of a cell through bubbles by creating more bubbles inside a bubble to represent the compartments. All cells have different compartments where different cycles and jobs happen. The hydrophilic heads and hydrophobic tails act as a filter along with the cell wall to regulate what goes in and out of the cell. These are called the membrane channels. We demonstrated this through the bubbles by laying a piece of string on top of the large bubble between the square straws. The circle of string represented the channel. We then stuck the straw through the string to show how things can enter through the channel. Only certain things can go through the string to reach the bubble, like how only certain things can enter the cell through its channel. When the right things enter the cell through its membrane channel, it creates a chemical reaction. This chemical reaction can play a key role in the origin of life.